Bismillahirrahmanirrahim. Assalamu alaikum. My name is Komal Ayoub, and I have done my MPhil in microbiology from Kohat University of Science and Technology. Today I'm going to present the research work which I have done in my MPhil. The topic of my research is mosquitocidal activity of indigenous bacillus strains isolated from the fields of district Kohat. These are the contents of my presentation, which I'm going to discuss today. Introduction, rationale, objectives, material method, rationale, and conclusion. First of all, I'm going to discuss the introduction of my research. Mosquito bites can cause skin irritation, causing the red bumps and itching, and it is very common in our daily life. But more than 80% of global population is at risk of mosquito-borne diseases. 1 billion people worldwide die from mosquito-borne diseases every year, and it has already registered in WHO in the year of 2013. Despite a well-established malaria control programs, 500,000 malaria infections and 50,000 malaria attributable deaths occur each year in Pakistan. Chemical insecticides are highly effective but have many adverse effects. Therefore, the use of biological control agents from the control of insects such as mosquitoes is a safer and sustainable strategy. Now comes towards the rationale of my research. Some of the bacillus species are known to have insecticidal activity against some insects, but there is no proper research to evaluate their activity against mosquitoes. Therefore, the current study is designed to evaluate the insecticidal activity of bacillus species isolated from the local fields of district Kohat against some stages of mosquitoes. These are the three objectives of my research. Number one was to isolate and characterize bacillus species from soil samples of district Kohat. Number two, to collect the mosquitoes and larvae from water and crops. And number three, to evaluate the mosquitocidal activity of those isolated bacillus species against mosquitoes. The study area of my research was agricultural microbiology lab of Kohat University. Materials and methods. The first step of my research was sampling. So I collect 7 to 8 samples of soil from the local fields of Kohat. Then I made serial dilutions of each and every sample. After that, I inoculated the serial dilution on media and incubated for 24 hours. For identification, I identified those isolates through colony morphology, microscopy, biochemical test, and after that, I molecularly identified those strain as well. As I told you earlier, the most important objective of my research was to evaluate the mosquitocidal activity. So for that, I have to collect the mosquito larvae from crops. So I collected mosquito larvae. After that, I centrifuged the bacteria broth of the strains which I have isolated earlier for the separation of supernatant and cell pellets for bioassay. Total 7 soil samples were probed for the isolation of bacillus species, and from those 7 samples, 3 bacillus strains were successfully isolated and identified through various biochemical tests. These are the images which I took during gram staining and spore staining. Bacillus species are gram-positive broad. You can see clearly over here. These are the tests. I have listed all those tests which I have done for the identification of those strains. Catalase test, starch hydrolysis test, BP test, straight test, motility test, indole test, and the last one is urease test. I have done many more as well, but I have listed these things. Okay, these are the images. You can see clearly the oxidase test positive and catalase test positive as well. Starch hydrolysis test, you can see the positive result, and citrate test. So for the bioassay, I have used 3 strains, 2 of Bacillus badomani and 1 Bacillus subtilis. These are the 3 strains which I have used for the activity. Okay. The results are shown over here in the table. You can see clearly. I have used three replicas of each medium. The supernatant and cell dilution are separated through centrifugation of broth and broth on the whole, as I use it as well. You can see clearly that the highest activity was shown in supernatant, then 71% in broth, and the least was in cell dilution. Same was shown in the other bacteria as well. You can see. Okay. Bacillus subtilis. Here you can also see that the highest activity was shown in supernatant, after that in broth, and the least was shown in cell dilution. The cell dilution is actually the cell pellet which was separated from supernatant. I diluted the cell pellet in distilled water and use it as a medium for the activity. These are the images which I have took during the activity. These are the containers which I have used for checking the activity, in which I have kept all these three mediums, and one for the control I have kept for getting the accurate result. Here you can clearly see the dead larvae of mosquito. This is the whole result. In this table, you can clearly see that the highest activity was shown by the bacterial strain of Padamani, FSL. After that, Bacillus species, and followed by Bacillus Padamani, PD10. 72% was shown by Bacillus Padamani, FSL. 70% was shown by Bacillus subtilis, and then 67%. Conclusion. From this study, it is concluded that the above mentioned strains can be used as an alternate of chemical insecticide against mosquitoes to control different mosquito-borne diseases. Thank you for your attention.